Special thank you to Google for this opportunity and congratulations for this meeting. Um, yes, it's a little bit mysterious, my talk, and this is from a journal club. And we know that the muscle atrophy, of course, it's very harmful in terms of the recovering, especially in athletes, but also in general population. But in the athletes, it's worse because the long time recovering of the total muscle function, it's very harmful for their careers. So about 6 years ago, 5 years ago, in a journal club with my students, um, I was show them this slide with the mechanisms of the muscle atrophy, and of course we are more interesting in this point of inducing muscle atrophy, the immobilization, the prolonged rest, and of course we are discussing all these mechanisms that are all very clear nowadays in the general biochemical mechanisms of the muscle atrophy. And of course, there is some damage of the muscle, some that are reversible, some we don't know if it is or not. And in the sequence of this journal club, one of my students asked me, what if you could prevent muscle atrophy? And it was a challenge, indeed. And at that time, I was working also with the industry of the supplementation. And I said, why not? We will go to try what a very common supplement in the exercise can made in this topic, this dynamic of atrophy and physical exercise. So this is how the idea of testing some that could prevent the muscle atrophy and related this with the physical exercise. Um, and the supplement was the BCAs, the branched chain amino acids, that we know that in all the process of atrophy, there are an increase in these BCAs by oxidation, mainly in the mitochondria. And of course, the amino acids are essential because in atrophy, as we know, there is an imbalance between the proteolysis and the synthesis of proteins. And of course, if there are some muscle damage and the damage of the fibers in particular of some types of fibers, uh, it's possible to induce regenerative mechanism that normally we perform with some physiotherapy and stimulating the muscle from the outside. And in these regeneration mechanisms, of course, that the myogenic satellite cells as a very important element, and it was our first aim, how these BCAs could influence satellite cells in order to prevent atrophy. The satellite cells that are quiescent in the normal muscle, and they express the PAX7 when they are activated and passes through several processes that included proliferation and differentiation, and then the regeneration in myocytes, and then the formation of myotubes and regeneration of the muscle. There are some markers that we used in this work, the PAX7, the MIF5, and the CMET, that are related to these phases of the satellite cells. The main importance in the BCAs that we have used is that first included also glutamine, and the proportion of the amino acids is 4:1:1. And normally in the market, and all the studies and the contradictions in the several studies that we have nowadays, this is because the supplement used is normally 2:1:1. So this is quite different from the others, not only by the inclusion of glutamine, but also because of this proportion. We have to use an animal model and a protocol of running in the treadmill, because in Portugal there's forbidden muscle biopsies for research purposes. And what we have done is first see what happens just with immobilization and exercise in the satellite cells. We have a treadmill protocol, progressive one in speed and also in the grade. And in a group of rats, we have immobilized on the right wind limb. We have another with the same immobilization that is following by exercise, and another group that is preceded with exercise, and then the respective controls. This is for 4 weeks. The daily of the animals was evaluated daily. And we use, I show just the results of the gastrocnemius, but we use two muscles, the soleus and the gastrocnemius. We have the cuts for staining with hematoxylin, eosin to see the integrity of the muscle, and then the staining with immunofluorescent antibodies for PAX7, MIF5, and c-Met, laminin for the membrane, and DAPI for the nucleus. And this is the result of the hematoxylin eosin, here the control. In the exercise, this is known, the inflammation-like that stimulates the regeneration of the muscle, some apoptosis of the cells and the newborn cells, and also a little thickness of the perimysium. However, in the immobilization rats, we could saw inflammation also, but fibrosis and intramuscular fat, what we have also confirmed with the red oil staining. When we saw just the outside of the muscle and we measured the ratio between the muscle weight and the body mass, we saw that after immobilization, the right limb, of course, is less muscle mass. When we previously perform exercise and then immobilized, this loss of muscle was higher, but immobilization after exercise, apparently they're recovering the mass of the muscle. However, the results that we obtained with the satellite cells was quite different. Here we saw, cannot see my slide, sorry. Okay. We saw that when we marked here the black for PAX7, MIF5, and c-Met, after immobilization, there is a clear known differentiation of the satellite cells. The exercise plus immobilization or the immobilization after exercise, it almost seemed the normal one, but immobilization after exercise, even we saw the mass of the muscle perfectly normal, the behavior of the satellite cells was not so regenerated as we expected. So just to look at the exercise without the supplementation, it seems that the exercise might have a protective or preventive effect against the immobilization and the damage that is caused by the atrophy. This suggests also alterations in the muscle architecture, and we have two distant inflammatory states. The exercise is well known. It's needed. It's a inflammatory like needed for the regeneration program of the myogenic cells. But in the atrophy, we have impaired in the muscle regeneration. Then we administered it in another group of the rats with the same characteristics, the BCAs, with the same amount or the same concentration that we use in human. And according to the body mass, it was an oral administration, just like the athletes or the normal exercise users take. And the protocol was the same. We just include immobilization by the supplement and the supplement plus immobilization, followed by exercise, 4 weeks of exercise. What we saw, the exercise immobilization was the same that I have described previously, but when we made the immobilization plus the supplement, it was the same as the exercise, some inflammation like and newborn cells. However, when we performed exercise after we administered the supplement and together with the immobilization, there's more severe damage in the muscle, and we have also the fibrosis. The same ratio, muscle height, body mass, we cannot see differences in the muscle from the outside, the macroscopy observation with the immobilization plus the supplement or following by exercise. However, it's just to remember the markers. And we obtain this type of staining here, the DAPI and PAC7, MIF5, and also the CMET here. And the result was this. We already saw this part in the previous slide. Now the immobilization plus the supplement, we see a higher regeneration in the satellite cells, so higher stimulation of the differentiation of the satellite cells. However, when we perform exercise after this protocol, we have the almost the reverse, a break in the regeneration of the, the muscle cells. So it seems that exercise may have a protective effect against immobilization damage. The effect of the exercise alone is similar to the supplementation with the BCAs. However, the BCAs has a high satellite cell stimulation. And for the other hand, the exercise after immobilization with BCA supplementation may inhibit the normal skeletal muscle regeneration. Um, so our work suggests that this supplementation with BCAs has a partial protective effect against damage by immobilization, but it seems to hamper the muscle regeneration when exercise is performed after the immobilization. So the BCAs might be very useful because they have influence in the muscle regeneration by acting over the satellite cells. Of course, I just show you a little bit of the results. We have a longer road to go in this work, but and before I finish, I would like to thank to also my students and my co-workers, and also the funds that we have for to perform this project. Thank you so much.
Thank you, Paula. Any questions?
I have a general question to the audience, but first to Paula. Which is the real significance to have more satellite cell activation? It is an evidence of more damage previously, or is an evidence of recovery? Because I am in doubt, still in doubt, because it is not easy to decide if you are looking 3 days later, 1 week later, to what happened a week in advance. This is, in my opinion, this is a basic open issues. And the next year, I hope we will have half a day to talk on these myogenesis regeneration issues from this point of view. Having evidence of regeneration, it is like reinnervation. If you have reinnervation, this means that you had denervation previously.
Yeah.
And this
Well,
is true also for the evidence of satellite cells activation.
yes.
What is your opinion?
We have results previously, during, and after. And yes, there is a higher activation during the exercise, for instance, only. And that is normal for me. It's an explanation that is related to what exercise is doing to the muscle. Because during exercise, we have a breakdown of the proteins of some fiber or myofibril structures. And it's natural that those substances that are releasing during the exercise will activate satellite cells. We have in this work the exact number before exercise, after exercise, and we can guarantee that is an activation for the damage caused by exercise. Small damage, of course, there are always a damage promoted by the exercise.
Did you do the embryonic and neonatal myosin heavy chain at the same time? Just
The
after?
embryonic, sorry?
Did you do the developmental myosin heavy chains to look at the regeneration at the same time? Because during exercise, you can just get activation of the satellite cells, but no damage, or you can have damage and regeneration, in which case the PAX7 cells would either be on the side or in the middle. There's a lot of things going on after exercise.
Yes, you refer to the histological.
Yeah, you could do. Have you done the immunology
yes,
with
yes.
the embryonic and the neonatal myosin heavy chains?
No, not embryonic and myomas. No, just to adult rats.
And are all the PAX7 cells in the satellite cell position?
Yes.
Yeah.
That the immunostaining was very clear on it.
Corrado? Microphone, please. Microphone.
I think this is a good suggestion because altogether they might do more exercise, provoke more damage, and they might have more, because of the glutamate that is somewhat excitatory, more exercise, provoke more damage. You need to monitor with CK and myosin embryonic, like Gillian Butler-Brown suggested.
Yep.
Any,
Stanley. No, you have a book on muscle damage, exercise-induced muscle damage. What about your opinion after 20 or 30 years? Microphone, please.
Stanley, use the microphone.
Microphone.
You're on mute. Ugo, you're throwing this off a little bit, but it's fine.
Okay, how about that?
Great.
It's very kind of you, Ugo, but that book was years and years ago.
Because this was a major issue in mass electrical stimulation. By electrical stimulation, you induce damage and therefore some recovery, over recovery, more recovery than damage. These are very interconnected problems. And the muscle dystrophy is the extreme examples.
Yeah.
Low level of exercise, a
I've
lot
used,
of damage, a lot of regeneration for
I've used
1, 2, 3
amino acids in dystrophy, but there was no effect, you know.
Yes.
I hope to get
Anyhow,
more
this was just to introduce the topic that we are ready to organize the next year Padua Muscle Days, whatever the COVID will be.